All right, I believe we're going to get started here. Thanks for coming and welcome to the Needham Healthcare Conference. I'd like to introduce Rachel King of GlycoMimetics.
Thanks very much, and thank you all for coming this afternoon. I'll begin by making reference to the fact that I will be making some forward-looking statements today. And to start the presentation, I'd like to begin by talking about our maturing clinical pipeline, which is leading to some important near-term catalysts. I'd like to mention several of our compounds, and then I'll go into a couple of these in more detail. First, let me highlight Rivipansel, which is currently in phase 3 for the treatment of vaso-occlusive crisis of sickle cell disease. We expect to have that data completed, the study completed and the data available mid-2019, so that's a readout that's coming up this summer. This is a program for which we had robust phase 2 results across all of the endpoints which we evaluated. Those were published in Blood. And we have a collaboration agreement now in place with Pfizer for this program. Pfizer now is projecting this as a potential blockbuster drug with possible peak sales of greater than $1 billion. Importantly, we have significant milestones due to GlycoMimetics from low double digits, and we have significant milestones, and we also have royalties due to the company from low double digits moving up to the low teens. We're also currently enrolling a phase 3 trial for uproleselan. This is our drug that's being tested for relapsed refractory AML disease. And we have breakthrough therapy designation received on this drug, which was granted to us in May of 2017. Here we're targeting top-line data in the fourth quarter, at the end of fourth quarter of 2020. And we have IP on this drug through 2032 in the U.S., in the EU, and in Japan. Importantly, with the uproleselan program, we also have an opportunity for market expansion beyond the initial phase 3 that we're conducting through 2 consortia-funded trials which are taking place, one being sponsored by the National Cancer Institute in the United States, and a second being sponsored by the HOVON Group in Europe. These are based on strong independent KOL interest, and these give us an opportunity potentially to expand the label into additional patient populations beyond the population that's specifically targeted in our phase 3. Specifically, we have an opportunity to go into patients who are newly diagnosed with AML, both patients who are fit for chemotherapy and patients who are unfit for chemotherapy. We have a strong balance sheet, which is funded importantly through that phase 3 readout with uproleselan. At the end of December in 2018, we had over $200 million in cash, and we're well positioned from this point to drive value creation going forward with the potentially game-changing set of therapeutic opportunities represented across our pipeline. I'd like, first of all, to dig into more detail on the Rivipansel program, which is the program that's targeting sickle cell crisis, and this is the program for which we anticipate a phase 3 readout in the middle of this year. Rivipansel works by targeting the selectins. It's a pan-selectin antagonist, and you can see here the cartoon of how the drug is believed to work by targeting the interactions between the white blood cells and the vascular endothelium. The selectins are expressed in the lining of the vasculature, and similar to the way Velcro would trap a tennis ball that's rolling by, the selectins interact with carbohydrates on the surface of the neutrophil, and they can slow the flow of neutrophils. Once the neutrophils bind to the vasculature, that activates a subsequent series of pathways which contribute to these vaso-occlusive events involving the sickled red blood cells, and that leads to the painful crisis, which is the specific event that we're attempting to treat in our phase 3 clinical trial. The selectins then activate these leukocytes, and as I said, trap the sickled red blood cells. So Rivipansel, as I said, is a pan-selectin antagonist. It disrupts the tethering and the adhesion of the cells on the endothelium, and we believe that it will be able to restore normal blood flow. In our animal models, that's just what we demonstrated, and the hypothesis has been going into the clinic that by improving the blood flow, by restoring the blood flow, we would be able to reduce the pain and then reduce the duration of the crisis. Sickle cell disease is one of the major single-gene defect diseases in the world. It's actually the most prevalent single-gene defect genetic disease in the United States, with approximately 100,000 people living with sickle cell disease. Importantly, it's also a serious condition in other major markets. Saudi Arabia actually has 145,000 people living with sickle cell disease. There are about 55,000 people in the major markets of Europe and 30,000 people in Brazil. So it does represent a very serious and important genetic disease one of the most common around the world. In our phase 3 study design, patients enroll when they're hospitalized, when they're actively experiencing a vaso-occlusive crisis. Then they're randomized to either get the standard of care plus ribapenzole or the standard of care plus a placebo. Thus, it's a randomized controlled clinical trial. We then follow the patients through the course of their hospital stay. We evaluate for a number of endpoints, which I'll describe some of the ones we evaluated in phase 2. Then the patients are discharged and they're evaluated one more time after they leave the hospital. Our primary endpoint in this study is time to readiness for discharge. This is an endpoint that we believe captures both clinical benefit and pharmacoeconomic benefit. Of course, the data will speak for itself when the data is available later this year, but we believe we have a high level of confidence as we look toward the ribapenzole readout, and we believe we can say that for a number of reasons. First, the selectins have been clinically validated now as targets for inhibition in sickle cell disease. We believe it's the only mechanism for which there are multiple controlled clinical trials which have shown actual clinical benefit, not biomarker benefit, but actual benefit on clinical endpoints. And I refer to our own phase 2 clinical trial. I'll share some of that data with you in a moment. That data was chosen for Best of ASH when we presented it. We were very pleased with the reception there. There was also a study that was conducted by Celexis, and this is a drug that's now being taken forward by Novartis, of a P-selectin antibody. And in addition to those 2 clinical trials that were conducted, there's further interest in this target expressed by the recent license that Global Blood took of a P-selectin antibody from Roche. So we believe that mechanistically we feel that it's been demonstrated that it's an important mechanism for targeting in sickle cell disease. Another reason for confidence is that the phase 3 primary endpoint is strongly modeled on our phase 2 data. As I said, the phase 3 endpoint is time to readiness for discharge. This is captured in a simple checklist of just a few items that are evaluated in the context of the trial. Things like, is the patient still on IV narcotics? Is the patient still on IV antibiotics? Is the patient off of oxygen? Things like that. And in addition, it's rigorously evaluated every 4 hours over the course of the study. So we think that it's a very well-designed endpoint that we think is well conceived to capture the benefit of the drug. The study is also well-powered. It's 4 times as large as the phase 2, and it's the subject of a special protocol assessment pre-negotiated with the FDA. This is the only drug in clinical trials for sickle cell disease that has a SPA. This gives us regulatory confidence as we head into the final data set. I want to highlight some of the data from our phase 2 trial. One of the key points in the phase 2 trial that's worth really driving home is that in the context of the phase 2, we had a very consistent data set that across clinical endpoints and across subpopulations that we evaluated, we saw consistent improvements of drug versus placebo. We thought the consistency of the readout in the phase 2 trial was really quite remarkable, and this is one of the things that gives us confidence as we move into the phase 3 readout. So here's, on this slide, you'll see 2 examples of data from the trial. One is time to resolution of VOC. This was actually the primary efficacy endpoint for the study. It was designed as a time to event analysis with the first of 3 events to occur, resulting in the capturing of the endpoint of resolution for crisis. Those events were either transition off of IV pain meds to oral in combination with a reduction in pain score, or readiness for discharge, or actual discharge. So depending on which occurred first, that was captured as the time to resolution of VOC, and you see the results graphed here. We saw a reduction of 63 hours. This did not reach statistical significance. But what we looked at next was we teased apart some of the things that went into time for resolution of VOC, and here we continued to build on the confidence in the data set as we looked at the various readouts in these other endpoints. One interesting readout was time to actual discharge from the hospital. This is a readout that can be confounded by many factors which can affect discharge from hospital. One example could be, let's say a child gets a discharge order written, but the parent is still working, and so they can't actually come to pick up the child for several more hours. So that's an example of the kind of thing that can confound time to actual discharge. Nevertheless, we did see a very meaningful improvement here of 84 hours with a trend towards statistical significance. Probably the most remarkable endpoint that we saw in the phase 2 is this effect that you can see on opioid use over time. Patients enrolled in the phase 2 trial were put on patient-controlled analgesia devices, enabling them to use the amount of narcotic that they felt they needed in order to treat their pain. And again, remember that this is a fully blinded, controlled clinical trial, so they did not know whether they were getting rivipansel or whether they were getting placebo. And here what you see is an 83% reduction in narcotic use during the period of the conduct of the trial, which was really a dramatic reduction in narcotic use. And you can see it here graphed as the average hourly narcotic use per 24 hours. And you can see that even in the first 24 hours, there was in that case, highly statistically significant reduction in the amount of opioid that the patients felt they needed to control their pain. So this is I would say, probably the most dramatic result that we found in the phase 2. And remember that patients are hospitalized because of their pain, and the reason that they have to be in the hospital is because they need to go on IV narcotics. So this is really a critical driver. Use of narcotics, use of IV narcotics is a critical driver not only of admission to the hospital, but also in the eventual decision to discharge. So that was a really critical and very exciting observation. We also looked at a related endpoint of time to transition to oral analgesics, and here also we saw a very meaningful improvement of 76 hours with a trend toward significance of 0.08. So in the aggregate, as I said, we felt that the results from the phase 2 were quite compelling, with a very consistent readout showing improvements across the endpoints and across the subgroups analyzed, and in some cases hitting statistical significance. This study is being conducted, our phase 3 is being conducted, as our phase 2 was, in patients hospitalized for treatment of vaso-occlusive crisis. Importantly, we believe there are expansion opportunities beyond that acute setting as we begin to look at potentially subacute treatment as well. In the phase 3 trial, we're treating these patients in the hospital. There are approximately 100,000 hospitalization events per year, so that's the primary market that we expect to be addressed with this drug. But in addition to that, as we look to the subacute market that I mentioned, we see that there are about another 150,000 events in which patients come to the hospital emergency room and are not admitted to the hospital. So while we think the drug, if successful in phase 3, could initially be used in that inpatient setting, we believe there are also some important opportunities to expand the use of the drug, looking first at the potential use in the emergency room. We also believe that the number of patients currently seeking care in the emergency room actually is a small, perhaps underrepresentation of the actual need for treatment of vaso-occlusive crisis in sickle cell patients. We say that for a number of reasons. One is that there is a lack of available alternatives to opioids other than supportive measures. So nobody likes to be on opioids, and the fact that is the only thing that can be given to really relieve these patients, we think, is a potential barrier to seeking care. We also believe that some of the emergency room attitudes that patients confront when they do seek care can be a barrier of care. Sometimes patients are thought to be drug-seeking when they come in and ask for narcotics. We think actually the data in our phase 2 study showed that that's not the case, because they used less drugs when they were on rivipansel. So we think that actually vindicates the fact that patients are using narcotics in order to treat their pain and not for other reasons. But we do know that there are some barriers to care that are seen in the attitudes that are expressed toward these patients. And one example of that is that we also know that sickle cell patients wait about 60% longer for pain meds than other patients, a real mark of their inability to get adequate rapid treatment for their crisis when they seek it in the emergency room in some cases. And further, that only about 20% of family physicians report feeling comfortable treating patients with sickle cell disease. It is a rare disease. There are a limited number of physicians who are specialized in it. And all of these things, we believe, contribute to the fact that likely what we're seeing currently in terms of the presentation at the emergency room is perhaps an underrepresentation of the need. And we hope that with rivipansel available, if it's successful, that we could more successfully enable these patients to get good care in the event that they're experiencing a VOC. So overall, we believe that rivipansel has the potential very attractive commercial opportunity, that there's a high unmet need, that this is unlikely to change in the future. This is an important opportunity in that it's the only on-demand treatment that's currently in late-stage development. We think that means it is potentially positioned rapidly to become a standard of care for VOC in the event that the trial is successful. Importantly also, there are similar treatment patterns, similar approaches to treatment in both the European Union and in the United States. That represents a possibility for global commercial alignment and opportunity to get into multiple markets. We also mentioned a sizable population in Saudi Arabia. There's also a fairly concentrated target audience. As a rare disease, the specialists tend to be concentrated, and geographically in the United States, the patients are concentrated generally in larger cities in the coast and in the southern part of the United States. So that makes commercialization possible with a small and focused sales infrastructure. And Pfizer now is beginning to talk about their view of the commercial opportunity, which they have described as a potential blockbuster for them with peak sales of greater than $1 billion. We have a partnership with Pfizer which gives substantial economics back to GlycoMimetics, and I highlight those here. At this point, Pfizer is fully responsible for the commercialization of Rivipansel. Our responsibility at GlycoMimetics was to get it through phase 2. At that point, we transitioned the responsibility to Pfizer. They're now running the phase 3 trial, and they'll be responsible for launch. As we look to upcoming milestones for this product, I can highlight a couple that are fairly near term. In particular, let me note first that we have about $150 million of fairly near-term milestones available to the company. These have not been broken out further than what you're seeing on the slide, but what I can tell you is that we have meaningful milestones now at acceptance of NDA and at acceptance of filing by EMA. We also have milestones at first commercial sale in the United States and first commercial sale in Europe. Those are followed by subsequent potential milestones based on Rivipansel achieving certain sales volumes. So we believe that these are meaningful milestones that are due to us potentially fairly near term in the event that the phase 3 is successful. And in addition to the milestones, we are slated to receive royalties on worldwide product sales, which range from low double digits to low teens. So in the aggregate, a very significant potential set of economics that could come to GlycoMimetics in the event that the product is successful. I'd like to turn now to a discussion of uproleselan. This is the drug that I mentioned, which is in clinical trials now for AML. We have received breakthrough therapy designation for this product, and we believe this also represents a significant market opportunity. In this case, a program that is wholly owned by GlycoMimetics. AML is a very significant unmet need. It has the highest incidence of leukemias among the leukemias and the worst overall 5-year survival. So it's a disease for which recently there are a number of drugs that have been tested and a couple approved, but it's one that over time has really, this overall survival number has really not moved substantially. And as you can see, with nearly 20,000 new cases in the United States, it represents the most common form of leukemia. The treatment landscape for AML is generally divided into 3 categories of patient, and those are shown here. First of all, at diagnosis, patients are described as being either fit for intensive chemotherapy or unfit for intensive chemotherapy, and they get different treatments depending on how they are described. Then if patients relapse or if they're refractory to the initial therapy, they fall into this category known as relapsed and refractory. So to simplify the landscape, we think of those 3 populations: newly diagnosed fit, newly diagnosed unfit, and relapsed refractory. Most of the clinical development now of new agents is actually taking place in that newly diagnosed unfit for chemo category. We're doing our initial trial, as you'll hear, in the relapsed refractory setting. But because of the extremely high interest from a number of KOLs around the world, we also have opportunities in the 2 newly diagnosed settings, which I'll speak to in a moment. So uproleselan works as an E-selectin antagonist, and what you see here is a diagram of the bone marrow where E-selectin is expressed constitutively on the lining of the vasculature. The AML blast has an E-selectin ligand on its surface, and that enables the blast to traffic to the bone marrow where it can adhere, where it can become sequestered and quiescent. And whereas with chemotherapy you can be treating the circulating tumor cells and often kill many of those, the cells that are sequestered in the bone marrow and quiescent are very difficult to kill, and those are then believed to be the source of relapse. So what we've shown in our preclinical models with uproleselan is that we're able to prevent trafficking of tumor cells to the bone marrow, that we're able to disrupt what is known as cell adhesion-mediated drug resistance. That's a phenomenon whereby cells bind to E-selectin and become more difficult to kill Even in vitro, they're more difficult to kill when they bind to E-selectin. We've shown that that's because binding to E-selectin upregulates tumor survival pathways, and we've shown that we can inhibit those pathways with uproleselan. We've also made the interesting observation that treatment with uproleselan in the context of chemotherapy in animal models protects normal hematopoietic stem cells from some of the toxicity of chemotherapy, a very interesting safety observation. And in addition, we've seen significant reductions in animals, and now we're beginning to translate this into humans, reductions in mucositis. That's the severe inflammation that occurs that affects the lining of the gut in patients who get these intensive chemotherapy regimens. So to summarize, mechanistically, what we think we have an opportunity here is to disrupt the interactions in the bone marrow microenvironment, make the tumor cells more susceptible to killing, and at the same time potentially to improve the underlying safety profile of the chemotherapy. It's really quite a differentiated and we think exciting set of attributes, potential attributes. One of the things that has really driven our interest in this target over the years has been the number of studies that have shown that E-selectin itself or the E-selectin ligand are correlated with worse outcomes in patients who have AML. And this was driven home again in a recent data set that was collected and presented at ASH this past year, in which Pam Becker from the Fred Hutchinson looked at a group of patient samples and analyzed them for E-selectin ligand and correlated that with clinical outcomes. And what she showed was that higher levels of E-selectin ligand were correlated with greater risk in disease. In particular, she showed that the E-selectin ligand was increased on patients with relapsed refractory disease. She also showed that the percent of binding was correlated with patients who had unfavorable risk as compared with favorable or intermediate risk, and showed that expression of E-selectin ligand also correlated with the markers of leukemic stem cells, suggesting that this is a mechanism for resistance, particularly for leukemic stem cells. So in general, we saw that the E-selectin ligand is correlated with chemotherapy resistance and with AML persistence. So what we're doing now with our program is trying to develop the clinical data set that will enable us to position this as a potential backbone treatment across a range of patient populations, so that across the continuum of care in AML, our goal is for this to be a foundational treatment. And what we're looking at now is, again, these three patient populations, and I'll touch briefly on each of these and let you know what we're doing with all of these populations. But conceptually, the idea is that we would be able to demonstrate synergy with the standard of care, so that we could use our agent in combination with whatever agent is the actual cytotoxic agent that's doing the tumor killing. Because our agent itself on its own is not cytotoxic. So it's an agent that needs to be combined with the cytotoxic agents, in each case with whatever the standard of care is with these patient populations. I'd like to share with you the data from the most recent data presentation that we had at ASH in December, just highlights from that presentation, in which we shared the data from our phase 2 study, which treated patients with both newly diagnosed patients who were fit for chemo and patients who are relapsed and refractory. So highlighting some of the data from that oral presentation. First, for relapsed refractory patients, what we showed was a 41% CR/CRI rate and an 8.8-month median overall survival. And I'll show you in a minute how we think that compares to relevant historical controls. For newly diagnosed patients, we showed that we had a 72% CR/CRI rate and a 9.2-month event-free survival. And EFS in this population is the endpoint that we're going to be looking at in our next clinical trial. We also showed, and this is an important new piece of data that we showed at ASH, we showed that greater than 50% of the evaluable patients achieved measurable residual disease negativity. In other words, by a very stringent measure, we were able to demonstrate that there was no detectable disease remaining in the bone marrow after treatment. In each case, greater than 50% of the evaluable patients. So these were small numbers in the phase 2, but in the phase 3, one of the things we're doing is gathering in a centralized way data on MRD negativity, as we will also be gathering data on E-selectin ligand and a centrally evaluated assay. And with respect to E-selectin ligand in the phase 2, what we showed was that it was detected in every patient tested, confirming the biological relevance of the target. And interestingly, I showed you in the previous slide that E-selectin ligand tended to be correlated with worse outcomes in patients not treated with uproleselan. In our phase 2, what we showed is that for those patients, when they were treated with uproleselan in combination with standard of care, they actually had better outcomes than the other patients. So we had higher response rates, we had greater MRD negativity rates, and prolonged median overall survival. So again, confirming the relevance of that target and confirming the potential benefit of treatment of that targeting of that with uproleselan. I want to show you some of the historical data to give you a sense of why we're enthusiastic about this data that we generated in our phase 2. First, looking at the relapsed refractory population, focusing on overall survival, which is going to be the endpoint for our phase 3 trial. You can see that in phase 2 we showed an overall survival of 8.8 months. And what we did now is to look back at the literature to generate a data set of demographically matched historical controls. It's quite critical in this patient population to ensure that our controls are from demographically matched populations. As you may know, looking at AML, there's quite a variation of outcomes depending on different demographics. So for this historical control evaluation, we looked through the literature. We found, in the case of the relapsed refractory disease, 3 studies where we felt that the populations were comparable and the treatments were comparable, so we can look at how those patients did. And there we saw 5.4, 5.2, and 3.4 month overall survival across those 3 studies. And then we looked for the newly diagnosed patients who were fit for chemo, similarly trying to find a demographically matched historical control population. And here we found 2 papers where we were able to evaluate the data on event-free survival and to compare it to the 9.2 months that we saw in our phase 2. And in these comparator trials, they reported 6.5 months in one and 2 months in the other. So again, we feel that the data was strong. It's very encouraging. In these 2 different patient populations, we'll be focusing now on the relapsed refractory population for our own clinical trial, and we'll be looking at newly diagnosed patients in the trial sponsored by the National Cancer Institute. So here, let me summarize where we're going with our next phase of studies. We're doing our randomized controlled phase 3. I'll show you a moment what the study design is for that. We've been very pleased to see the high level of interest from KOLs around the world in the data set that we generated in the phase 2 study, and that has enabled us now to go into 2 consortia-funded trials, which position us potentially to expand the label in a very cost-efficient way by working with these 2 other groups to test the drug in 2 additional patient populations. So in parallel with the study that we're running in the relapsed refractory population, the National Cancer Institute is sponsoring and funding a clinical trial in newly diagnosed patients fit for chemo, and the HOVON group in Europe is sponsoring and funding a trial in newly diagnosed patients who are unfit for chemo. So if you think back to the slide I showed at the beginning where I described the 3 different populations of patients in the AML treatment landscape, we are now positioning the drug in all 3 of those populations with our own funded trial and these 2 consortia-funded trials. So we think it gives us a very cost-effective way for us to get a lot of visibility around the trial, for us to engage KOLs around the world, and potentially to get to a broad label very efficiently. I want to summarize this section of the presentation by showing you the design of the phase 3 as we now move forward with that trial. That trial is currently already enrolling. We've enrolled patients in the United States and in Australia, and we expect shortly to add a number of sites in Europe where we will also be enrolling patients. So patients get randomized, and they get randomized to receive either chemotherapy plus upro or chemotherapy plus placebo. And after that induction cycle, if they respond, they would continue to potentially up to 3 cycles of consolidation, again with chemo plus upro or chemo plus placebo. So we believe that if our drug is effective, being able to treat patients with multiple cycles continuing on that protocol enables us to potentially differentiate outcomes even further. Most of the patients in our phase 2 trial only had 1 cycle of treatment with uproleselan, so we think this is an important advantage now as we move into phase 3. And then the patients will be followed for overall survival. So to highlight our pipeline, in addition to the rivipansel drug, which is in phase 3, for which we have the readout coming shortly, and in addition to the uproleselan, which I've described in AML, we have another program, 1359, which we're carrying forward Currently, we've completed phase 1. We'll be saying more shortly about the initial patient trial that we anticipate there. We've done a number of preclinical studies in different tumor types and presented them at a series of major scientific meetings, so we'll be saying more about that program later this year. And we also have a very exciting preclinical program in the galectins. This is a novel target, one that's involved both in cancer and fibrosis, where we're currently generating a number of very active molecules. And I'll highlight one other compound which we announced at ASH this year, and that is still a preclinical compound, but one that is highly potent and potentially subcutaneously available, which could give us an opportunity for a follow-on drug to uproleselan. We have a number of important announcements anticipated in the second quarter. One is the initiation of enrollment in the National Cancer Institute-sponsored trial. A second is the potential initiation of the HOVON-sponsored trial in Europe. But most importantly, as we move into the summer, looking for the output from the rivipansel phase 3 study, we expect that in the second quarter we should be able to announce completion of enrollment in that trial, and that over the summer we should be able to announce the top-line data for that study. So to conclude, we have, I think, a very exciting pipeline of opportunities which we're well funded to deliver on. We have, first of all, rivipansel, the phase 3 program being developed now by Pfizer for treatment of vaso-occlusive crisis of sickle cell disease. We have GMI-1271, now known as uproleselan, for which we've received the breakthrough therapy designation for the FDA, currently in phase 3 trial, sponsored by the company, as well as these other trials that will be sponsored by these collaborative groups. And we have 1359, for which we expect to announce additional plans later this year. Both rivipansel and uproleselan represent important market opportunities for the company through our milestones and royalty opportunities with rivipansel, through potential commercial sales by GlycoMimetics itself for uproleselan. And we have a strong investment base represented by some top-tier biotech investors. As of the end of 2018, we had over $200 million in the bank, so we're well funded to continue to drive these programs. And we're led by a team of experienced executives in glycobiology as well as in biotech generally, and we have the benefit of important and key relationships with KOLs around the world. And so I'll conclude there. Thank you very much for your attention, and take any questions that anyone may have.
With your discussions with Pfizer for example, do they have any thoughts about pricing and payer relations? Because I'm assuming that can take care of that.
Yes, so Pfizer has not said anything publicly about pricing, and we know that they're actively actively doing the pre-launch planning that you would expect as they lead into the potential phase 3 readout that's coming for that. The only thing that they've said publicly to give us a sense of how they think of the scope of the opportunity is that they have described it as a potential blockbuster within their own pipeline, and that to them that means peak sales of greater than $1 billion. So with 100,000 hospitalizations in the U.S. only, you don't have to think about pricing being terribly aggressive to get easily to $1 billion. So I think you could run a series of potential pricing scenarios as you think about how to get there. But it's certainly helpful for us as we characterize the opportunity that Pfizer is now describing it for them as a potential blockbuster. But they have not given any specific indication around pricing, and I don't expect them to do that until closer to launch.
As for the cancer drug, safety as it appears, it should be added to literally every other AML treatment out there, targeted, untargeted, doesn't really matter. So there's a lot of potential for combination.
Yeah, that's exactly right, and that's exactly what you see reflected in the development plan that we've outlined so far. The plan that we're currently implementing combines with 4 different chemotherapies, one for newly diagnosed fit, another one for newly diagnosed unfit, and then 2 different chemotherapies used in the relapse refractory setting. That's the start of the program. That's not the end of the program. We've had a great deal of interest in doing further combinations that many investigators have approached us about looking at other possible ways to study uproleselan. It's because the mechanism of action is so differentiated, there's actually a very sound biological rationale to potentially combine it with multiple agents that goes beyond simply, 1 plus 1 may be more than 2. It's actually a potentially synergistic approach that we could take by combining with these cytotoxic agents. So yes, that's exactly true, and that's what we're trying to do with our development plan. Yes, it is, and we've had some very interesting and encouraging preclinical data in multiple myeloma as well. We did initiate a small trial in multiple myeloma in Europe, which we have discontinued because the myeloma landscape has changed so much with the approval of daratumumab that we didn't see an opportunity, we didn't see as great an opportunity there as we see in AML. So we've decided that it's better for the company and for the program that we would devote our drug supply and our resources to expanding the use of uproleselan first within the AML landscape, and then from that point we'll look forward to other potential indications. But at this point we decided not to continue to pursue it in myeloma, although it really did show in preclinical models that there was a very strong rationale for potentially using it in myeloma.
I find that actually a little surprising considering AML, you get MRD resistance and so on, curative, and multiple myeloma, there's always a deep-seated clonal population.
Yes, and so I do think that there's a clinical opportunity potentially to show benefit with uproleselan. What's challenging is in the actual real-world treatment landscape where you've got so many drugs in so many lines of therapy that are well established in the treatment of myeloma, it's difficult to actually implement a clinical development program. And for us, we just felt that there's greater opportunity in AML at this point, even though there is a strong biological rationale. Great. Thank you very much.